Hello and welcome to the Ornithology Department. Come on in. So here we have our workspace. The Ornithology Department at the Natural History Museum of Los Angeles County, we share space with mammalogy, so you might see a few mammals around. But we're going to really be focusing on the birds here. So let's walk through the workspace. We have, of course, some offices. We'll show you our digitization room. This is where you can make 3D scans of specimens or take photos with a UV vis camera because birds see into the ultraviolet part of the spectrum, so it's important to capture both. Here is our work area, but really the star, of course, of the collection is our where we keep our specimens. So let's come on into the main range. Now here in our main range, you can see it's very grandiose because we have over 100,000 bird study skins, and mammals has, I think, about 90,000 mammal skins here. We start with mammals. So we're just going to pass by all those mammals because, of course, we are focused on birds. So here's where it ends. So mammals from here on down, birds from here on down. If we go into one of our aisles, you can see that we've got the cases of specimens are stacked 3 high. You can imagine that 100,000 bird skins take up quite a bit of space. Now we can open up one of these cases, look inside, pull out a drawer, and every one of these cases has specimens lined up in rows just like this. Now these you might recognize are red-tailed hawks. See, this one has a very nice red tail. Sometimes it's actually really nice to have these specimens in hand so you can see the details that might be really difficult to see in the field. But one thing you might notice is that these specimens are not prepared to look lifelike. You might think of when you come to the museum, you see birds that are taxidermied in different poses. Now think about if we had 100,000 specimens prepared to look lifelike instead of prepared like you see here, they would take up a lot more space. And so there are, in addition to space constraints, there are reasons that you can go to any museum anywhere in the world, open up a case, and specimens will be prepared just like this. So from a consistency perspective, we can take the same types of measurements, we can do the same types of analyses, and they're also consistent over time. Now, one question that I get very commonly asked as curator is, why do you need so many specimens? Why do you need 100,000 specimens? There are about 10,000 species of birds. We have about over half of them in our collection. But this drawer is just red-tailed hawks, and in fact, almost this entire case, as well as this drawer, are red-tailed hawks. And just looking at this drawer, you can start to answer that question. Well, you might notice that birds are different colors. So we have birds that have adult plumage, we have birds that have juvenile plumage or immature plumage. We actually have this melanistic individual that's got very dark plumage. So you can see there's a lot of variation that exists. You might notice variation in size. Compare this specimen to this specimen. You can notice that it's a lot bigger. This one's actually a female, this one's a male. Those are some of the easier differences to see by eye, but if I measured the size of the beaks or the size of the claws or the size of the feet or the length of the wings, there would be some small differences between different individuals. And so it's really that variation that makes it necessary to have more individuals. You might also notice that there's a little tag tied to the foot of every specimen in our collection. And without that tag, these specimens would become virtually useless. So you can see that this individual is from Orange County, from Seal Beach. It was from 18th of January, 2013. You can see there's a note that one feather louse was saved. So we have a whole nother collection of parasites and things like stomach contents that we actually can link to individual specimens. We've got a prep number, and there's going to be even more information about this individual in our digital catalog. So all of the data on all of the tags are all digitized and available online. You can go to a website called GBIF, gbif.org, search our collection and see exactly what we have and where it's from and when it's from. So if I was to compare these localities on these tags, you would see that some are from Orange County, but you might notice that others are from other parts of the world or parts of the country. So this individual is from Palms. Actually, that's from pretty recent time, but it's from 1927, December 4th, 1927. And so you can start to notice that in addition to just variation between, males and females, adults and juveniles, there also might be geographic variation. So birds from one area might be a little bit different than the others. And so we need representations from different geographies in our collection. And then there's the axis of variation. That means that we are never going to be done growing our collection, which is time. And so each of these specimens is from a specific date. And it means that these are actually little time machines to that point in space and time. So I can take DNA, I can take a little skin sample from the foot of any of these birds, and I can sequence their genomes, and I can understand from a genetic perspective what were birds like in that place and time. We can then compare them to modern birds and understand, have birds evolved over the last 100 years? You think of evolution as taking, thousands of years or millions of years, but in fact, evolution could happen really fast, and we're learning that with these historical collections. I might take a little feather sample and try and see if there were any heavy metals in the feathers and understand, were these birds being exposed to toxins back in that time, and how has that shifted over time and space? I can look at bill size change or wing length changes or body size changes. But having each of these little time machines allows us to understand, how are birds responding to environmental changes? Just like smoke, like we're looking at with Project Phoenix. We actually can analyze, how much black carbon is in these feathers and try and understand something about what sorts of air pollution, which, was it due to industrial reasons or wildfire smoke? How has that impacted birds through time? And combine that with the wonderful observations that you have all submitted to understand, how has that impacted birds in the past, and how are they responding now, and how can we help them? So each of these specimens is incredibly valuable. Now the way that we've collected specimens has changed. So almost all of these specimens have come now from wildlife rehabilitators or birds that people have found deceased. So, I might get a call that, hey, I found a red-tailed hawk in the street, would you be interested in having that for your collection? So you could email me if you ever find a dead bird, and that's how we are growing our collection these days. Now we'll just show you a few more cool birds. So in addition to, those are fairly big birds, but probably some of the biggest birds that we have in cabinets in our collections are these. This is a California condor. Look at the size of this thing. Turkey vulture is probably about this big. This individual is from 1923, and so, from a conservation perspective, we can take some skin samples, sequence their genomes, and understand, when condors were almost lost to us, how much genetic diversity was lost through time. And you never know with as technology progresses, how maybe we can even start to introduce some of that genetic diversity back into the population. But without any of these specimens, all of that would be essentially impossible to do. So we're going to go over here. We'll look at another cabinet of birds. So in addition to big birds, of course, we also have small birds. You might recognize here some of our friendly house finches. You may have seen these in your survey. Many of these house finches were from the early 1900s and can demonstrate really well how we might observe birds that are, maybe look a little bit dirtier than they used to. And so we have some research going on to try and understand how that's changed through time and what were the sources of that black carbon. In addition to common birds, we also have birds from other parts of the world. So here are some different finches from places in Europe and Asia. Now, birds are arranged taxonomically, so the closer you are in the collection, here's some evening grosbeaks, which are incredibly beautiful. The closer you are in the collection, generally the more closely related you are. Now, that's not 100% true because what we know about who's related to who has changed quite a lot since kind of the genomics revolution, and we still have to rearrange our collection. You might imagine that's a pretty big project. Here's some other French relatives. These are some pretty cool ones. You can see these are crossbills. So you can go up to the mountain, up higher elevations, and see these birds that specialize on eating the seeds of pine cones. They've got these bills that are crossed over each other. Now, different collections have different specialties. So of course we have many birds from California in our collection, given our geographic location. But back, and especially in the 60s and 70s, the museum went on expeditions to different parts of the world, especially to collect animals for our African mammal diorama halls. But while they were doing those expeditions, they also would bring back, thousands of birds and mammals. And so we have now very good collections of birds from East Africa. We also have very good collections from Brazil, from Colombia, from Argentina. And different museums have different specialties, kind of just depending on their history. These are starlings. I should have mentioned that earlier. So you might think of the European starling that we have here, which are, of course, actually quite beautiful birds, but other starlings show this beautiful iridescent color even more. Here we've got our friends, the European starlings, which again, quite beautiful once you look at them very closely. So now here we are in our prep lab. This is where we prepare new specimens. So they're not just kind of, specimens from a long time ago. We also have modern specimens. So here we've got all of our recently prepared birds. You might notice that we have all of these wings out. So, historically, people used to prep birds where both of the wings were against the body. And to prep them, what we do is we'll cut an opening. We'll take out all of what I call the goopy bits, so all the muscle, all the organs, anything that's going to be rotten. We'll leave the wing bones, we'll leave the skull, we'll leave the foot bones, and we'll stuff them with cotton. And then we just pin them out on these boards to dry. This, might take a week, it might take a couple of weeks, but once they're dried, essentially they're done. We stick them in the freezer to ensure that there's no pests associated with them. But then they go into we'll put them in our catalog and just put them in the collection. So, no, there's actually not, I won't say that it's easy to prep specimens because it's quite a kind of an art almost, and a skill that needs to be learned, but it's not actually that technical, and it hasn't really changed that much since the last 100 years. One difference is, in addition to like a skin, now we're cutting off one wing and pinning it out so that you can see the flight feathers. We have a whole wing collection that's associated with these birds. We also have a cryogenic freezer that we keep at -80 degrees, where we'll take samples of different muscles and different organs so that they're easier to do different types of genetic analyses. So a single specimen might have several different parts in different part of the collection. If they've got parasites, they might live in parasite vials. If they've got stomach contents, they'll live in stomach content collection. And all of that is tied together with our digital database. Now we're gonna go into our alcohol, our skeleton collection. So in addition to those 100,000 steady skins, we also have about 3,000 fluid preserved specimens. So fluid preserved specimens, think pickled bird in a jar, just like you think. Now the advantages of these specimens are they have all of their insides still. So if I want to study their muscles or study something about their organs, basically I need one of these because all of that is taken out of the steady skins. And it used to be that these weren't very heavily used because you would need to dissect out whatever you want. But with modern technologies now, these are becoming increasingly important and increasingly popular because you can just take this bird out of the jar, dry it off, stick it in a CT scanner, and get 3D reconstructions of everything on the inside of this bird. So, one thing we like to talk about as curators is we don't know what technologies are going to be here in the future, and so we have to preserve everything we can. And these physical specimens allow us to get information that we didn't even know they wanted to do. 100 years ago, they didn't know what DNA was, and now that's probably the top use of our specimen collection, is to get genetic material. So who knows, another 100 years, what people will want and what they'll be able to do. Now we're gonna just move a couple of these aisles. So in addition to our about 3,000 fluid preserved specimens, we actually have about 17,000 bird skeletons. We've got one of the largest bird skeleton collections, especially in the western US, and that's because of the La Brea tar pits and our amazing paleontology programs that we have here. You might think of birds as the tar pits. How do you know what's in there? Well, you have to compare it to something. And so we have a lot of the material of birds that you can compare it to understand what it is. From our paleontology department, you might need to look at bones. So you can understand, how bones are associated with modern birds, and then associate that with other birds. These are the bones of a hornbill. You might notice that bird skeletons are not just like the specimens. They're not prepared to look lifelike, but instead are prepared to be very space efficient. So bird bones in a box. As a researcher, I don't actually want to have something that's all put together, because I want to get whatever bone of interest I'm interested in. I want to be able to photograph it, to 3D scan it, to measure it, whatever I need to do to this bone, and I want to compare it across individuals. This is a hornbill. It's just very cool to see the inside of a hornbill skull. All that. Now these bones are incredibly light because bird bones are hollowed out, because birds are all about being as light as they possibly can, because they need that for flying. So yeah, so with that, we've got about 124,000 birds in our specimen collection. Increasingly useful for different things. As birds as time machines to the past, I would say, is increasingly being how the collection's being used, instead of just to understand evolutionary relationships. And we're happy to take any questions in our live Q&A. Thank you for listening.